Welcome back everyone. Today we are going to discuss this important topic under the cellular physiology, that is cytoskeletal filaments and the cellular junctions. So this cytoskeletal, first we are going to discuss this cytoskeletal filaments. These cytoskeletal filaments are called the bone of the cells. The cytoskeletal filaments, from the name we can know that they are called the bone of the cells. It's required for strength and support, also combined with the cellular movements of the cell. The cytoskeletal filaments are important for the strength and support as well as the movements of the cell. So we're going to see the classification, classified depending on the size. Depending on the size, they are of three types. There are the three types we're going to see now. First are the big filaments. These big filaments are called microtubules. These big filaments are called microtubules. Then there are the small filaments. Small filaments are called small microfilaments or simply microfilaments. The third is the intermediate filaments. These intermediate filaments are the, remember, most abundant ones. Most abundant ones are the intermediate filaments. The big filaments are called microtubules, not to be confused with the word big and micro. These are named in such nomenclature, we can't do anything. The big filaments are called here microtubules. The small filaments are simply named microfilaments. Then the intermediate filaments, which are most abundant. First we are going to discuss this microtubules. Microtubules are the three structural proteins with three functions. These are three structural proteins with three functions. What are the structural proteins? The first is kinesin. Second one is dynein, dynein. Third one is tubulin. Three structural proteins are kinesin, dynein and tubulin. These microtubules are also called as, change the color, force generating proteins. Force generating proteins are the microtubules, which are three important structural proteins, which are kinesin, dynein and tubulin. So we have to remember kinesin, dynein and tubulin. Now we are going to see the functions. The first function is in the neurons. In the neurons, what are the functions? Function is for axonal transport. These are involved in the axonal transport in the neurons. That is from cell body to the synapse. We know in general nerve physiology, this is the cell body. This is generally the axon, and this is the synapse. Cell body, axon, synapse. So in general, the axonal transport is from the cell body towards the synapse. From the cell body towards the synapse. Which protein is involved in this? Remember, the kinesin protein. The kinesin protein is involved in this axonal transport from cell body to the synapse in the neurons. Now we are going to see an exception to this. The exception is called the reverse axonal transport. As the axonal transport is from cell body towards the synapse, this reverse axonal transport is from synapse towards the cell body. From synapse towards the cell body. This is the reverse axonal transport. So why this reverse axonal transport occurs? First we are going to see the protein involved. The protein involved is the dynein. The dynein protein is involved here in the reverse axonal transport from synapse towards the cell body. This phenomenon of reverse axonal transport, is used by microorganisms. Some examples we are going to see, like the rabies virus or the herpes virus, also the tetanus toxin. Rabies virus, herpes virus and the tetanus toxin. Microorganisms involved in this uses the reverse axonal transport for their infection and further spreading. First function we have seen in the neurons. Next function we are going to see, that is function of this dynein protein. This dynein movement, as you have seen, this cytoskeletal filaments are also involved in the cellular movements. This dynein movement of three important structures, clinically very important this topic to see. Three important structure. First, in the lungs there is movement of cilia. So we know that there are presence of this ciliated columnar cells. Ciliated columnar cells contains this small cilia. Above these cells there occurs accumulation of this debris. This debris accumulate above the ciliated columnar cells, thus barring the movement of cilia. These debris are called sputum. Sputum or the debris accumulates above the ciliated columnar cells, thus barring the movement of cilia. This movement of cilia is towards the nasopharynx. It's towards the nasopharynx to expel this sputum, this debris or sputum which gets accumulated. The movement of this cilia is important because it is towards the nasopharynx, which is to expel this sputum. It's a protective function. This movement of cilia is a protective function. It's synchronized ciliary movement, synchronous ciliary movement is called metachromism. Metachromism, which is due to, we've seen above, the dynein protein in the cilia. Synchronous ciliary movement, which is important for expelling the sputum, is called metachromism. This is due to the dynein protein. So this is one function of the dynein protein in the lungs, or we can say more specifically in the respiratory tract, in the expelling sputum. Second one we are going to see is in the male germ cell, that is the sperm. The sperm movement requires this dynein protein. First is the lungs, the cilia, and this male germ cell, that is the sperm movement requires the dynein protein. Then the third is in the fallopian tubes. Fallopian tubes, we know there is also presence of cilia for the movement of both ovum and the sperm for processes such as fertilization and then further implantation and in the further development of the embryo and the fetus. This fallopian tube also requires dynein. This dynein we've seen is important in the fallopian tube, in the male germ cell, in the lungs, in the cilia, and also in this reverse axonal transport. It is used by these microorganisms to infect and spread. So this absence of dynein, remember, leads to immobile ciliary syndrome. This absence of dynein protein leads to immobile ciliary syndrome. It is also clinically known as this important syndrome, Carter-Jenner syndrome. Carter-Jenner syndrome, which is the immobile ciliary syndrome due to absence of dynein protein. So we have seen the functions of the microtubules in the neurons and specific functions of dynein protein. Third we are going to see is the tubulin protein. First we have seen in the neurons the kinesin protein, then the importance of the dynein protein. Third we are going to see tubulin. This tubulin protein is required during cell division. That is, mitosis. It's required during the mitosis. How it is required? It is required during the movement of chromosomes. Movement of chromosomes to daughter cells for survival of the cells. We know this movement of chromosomes is an important phase in this cell division or mitosis. This movement of chromosome to the daughter cells requires this tubulin protein. The tubulin protein helps in this process of movement of chromosomes. So seen these three microtubules. So we have some microtubule inhibitors for treatment purposes, pharmacology. We have some microtubule inhibitors. These microtubule inhibitors causes no chromosomal movement As the tubulin protein is involved in this movement of chromosomes, this microtubule inhibitors causes no chromosomal movement. This is important in cancer cell death treatment. This treatment of the cancer cells, as this movement of chromosomes is required for the survival of the cells, so no chromosomal movement can lead to certain cells death. In this case, cancer cells death, which often is used in the treatment of cancer. Since some examples of these inhibitors are the Wins alkaloids. For example, vincristine, vinblastine. The Wins alkaloids, the vincristine and the vinblastine, are often used as the microtubule inhibitors. For the treatment of gout, we need to prevent the movement of neutrophils. In general medicine, we're going to learn about this gout. Also in the biochemistry section, we are going to learn much about this gout. So remember, this movement of neutrophils is an important phase in the development of gout. To prevent this movement of neutrophils, we use treatment as colchicine. Colchicine is used in the treatment of gout to prevent the movement of neutrophils. So with this, we conclude the discussion of microtubules. Next, we are going to see are the microfilaments. Microfilaments. These are the small filaments, as we have seen in the classification, the cytoskeletal filaments. These are the microfilaments or small filaments, small cytoskeletal compounds. These are two structural proteins with two very important functions. It's two structural proteins with two important functions. As you know from the name, you can guess what are they. Yes, they are the actin and the myosin. Are all general actin and myosin in the muscles are examples of these microfilaments. So actin and myosin are the microfilaments, especially in the muscles. Now we are going to see the functions of these microfilaments. As usual, in the skeletal muscle. Muscle contraction and relaxation. Obviously, they are involved in muscle contraction and relaxation by this important sliding filament theory. We're going to discuss this theory in details in the dark muscle physiology. So this actin and myosin in the skeletal muscles are involved in the contraction and relaxation by this sliding filament theory. Next function, it plays an important role in cellular movement. Plays an important role in the cellular movement. How? Then we see this actin polymerization. This actin polymerization is a process in the cellular movement. This actin polymerization is often hijacked by this microorganism Listeria. This organism Listeria hijack the actin polymerization process, thereby developing a structure called poly-A-L. A stands for actin. And this Listeria hijacks this actin polymerization process, it develops a poly-A tail. This develops into a pattern called tumbling motility pattern. So as usual, actin polymerization if hijacked by listeria, the motility patterns get altered. General cellular movements, the patterns get altered to a tumbling motility pattern. So next I'm going to see, we have discussed the microfilaments, the actin and myosin, their roles in skeletal muscle and in cellular movement, and it is hijacked by this listeria organism. Next we are going to see the intermediate filaments. Intermediate filaments. These intermediate filaments are very specific. This is the first point we need to understand. Intermediate filaments are very specific. They are not found in each and every organ or cell. It is very specific to specific cell types. As they are very specific to cell types, these are often tumor markers. These intermediate filaments, for their specificity, are often the tumor markers. First intermediate filament we are going to discuss is the keratin. This keratin protein, we know, is present in hair. It's a very important structural protein. This keratin is expressed in epithelial cells. This keratin is expressed in the epithelial cells, which is considered tumor markers, a tumor marker of carcinomas. The carcinomas, the tumor markers, for example, cytokeratin. Cytoskeleton is a tumor marker for squamous cell carcinoma. Cytokeratin is a marker for squamous cell carcinoma. This keratin is often expressed in the alcoholic liver diseases when there is this upon cytokeratin accumulation. Cytokeratin we've seen here is a tumor marker of squamous cell carcinoma. In this alcoholic liver disease, there is cytokeratin accumulation in the form of these important structures called Mallory-Denk bodies. Mallory-Denk bodies. This alcoholic liver disease is cytokeratin accumulation in the form of these specific structures called Mallory-Denk bodies. It's an important pathological finding while diagnosing the alcoholic liver disease. Next intermediate filament we are going to discuss is desmin. This desmin protein is present in muscles. This desmin protein is present in muscles. It's also a tumor marker for sarcomas. For example of this condition, rhabdomyosarcoma. rhabdomyosarcoma, myo for muscles, sarcoma, tumor marker for sarcomas, cancer of the sarcomas is derived from this sarcomere, which is a structural unit of every muscle fiber. So desmin in muscles is a tumor marker for sarcomas. For example, rhabdomyosarcoma. Third we are going to discuss is vimentin. This is often expressed in connective tissue cells. Before we have seen this keratin, it is expressed in the epithelial cells. Now this vimentin is expressed in the connective tissue cells. For example, in the fibroblasts. Fibroblast is an example of connective tissue. In the cells of fibroblasts, vimentin is expressed. It's a tumor marker for mesenchymal origin. The carcinomas that are of mesenchymal origin, this vimentin is a tumor marker expressed in the connective tissue cells such as fibroblasts. Next we are going to see this important astrocytes. Very common familiar name is astrocytes. This astrocyte is an intermediate filament. We have this protein called GFAP or glial fibrillary acidic protein. Glial fibrillary acidic protein, GFAP. This astrocyte is this specific protein is a tumor marker of gliomas. That is the carcinoma of the glial cells. There are different glial cells in the central nervous system. We are going to discuss in the subsequent videos. It's a tumor marker of the gliomas. One of the glial cell is this astrocytes. For example, astrocytoma. This astrocytoma, it is a type of glioma. This astrocyte is a tumor marker. Fifth intermediate filament we are going to discuss is lamin. Expressed in the nucleus of cells. Expressed in the nucleus of the cells. This lamin mutation can lead to premature aging. This premature aging can occur due to mutation of this lamin, which is expressed in the nucleus of the cell. You know, nucleus is the controlling center of the cell. This mutation will lead to premature aging. It's called Werner syndrome or progeria. This Werner syndrome or progeria is, you can say, form of premature aging. It is the subject ages very prematurely due to mutation in this lamin, which is expressed in the nucleus of the cell, which is the controlling center of the cell. Werner syndrome, progeria. So these are all about the types of cytoskeletal filaments, the intermediate filaments, the microfilaments, and the microtubules. We have seen different proteins, the subdivisions, and their clinical conditions, the diseases, how their mutation or even absence can cause them. Now we are going to discuss this topic. after the cytoskeletal filaments is cellular junctions. Discuss this cellular junctions. A cellular junction. First, we are going to see simple diagram for understanding. This is cell. This is another cell. Cell 1, cell 2. This junction is called a cell junction. Between two cells there is a cell junction. This is the first type, between two cells. Second, we are going to see this is a basal lamina. It's a junction between the cell and a basal lamina. I call this as cell basal lamina junction. For simple understanding, there are two cells. In between two cells, there is a junction. It's called cell junction, obviously. And there is a junction between the cell and the basal lamina. So basal lamina, above which the cell rests, is a junction between these two. It's called cell basal lamina junction. There are two types of cellular junctions. We are going to discuss one by one. First is the cell junction. Cell junctions. We're going to see three types of junctions here. I need to draw first cell 1, and it's a second cell 2. First junction we're going to see is this one. It's cell adhesive junctions. Cell adhesive junctions causes adhesion like a glue between two cells, cell 1 and cell 2. Next type of junction we are going to see, this form of junction is called tight junction. The second type of junction between two cells, this tight junction. The third type of junction we are going to see here is this is gap junction. It's a gap junction. So you've seen here three types of cell junctions: cell to cell junction, cell adhesive junction, tight junctions, and gap junctions. So first we are going to see cell adhesive junctions. Cell adhesive junctions. Cell adhesive junctions uses cell adhesions or glue. This adhesion is what represents gluing of two substances each other. It uses cell adhesions. Cell adhesions molecules. What are these molecules? For example, cadherins. Second is integrins. Cadherins, integrins. The third one is selectins. Cadherins, integrins, and selectins. These are the cell adhesion molecules used by the cell adhesive junctions. So we now see a generalized pattern of this cell adhesive junction. So there is first a primary protein. Okay, so first is a primary protein, which is then attached to a linker protein. This primary protein is attached to a linker protein, which is then further attached to the cytoskeletal filaments. These linker proteins are then attached to the various cytoskeletal filaments. So this is a generalized structural arrangement of cell adhesive junctions. So there are two types of these cell adhesive junctions. Two types. First is the zona adherence, cell adhesion, zona adherence. Very simple understanding. Zona adherence in the epithelial cells. Zona adherence in the epithelial cells. So we are going to see how they are arranged. We have seen this. There is a primary protein. This primary protein is cadherin. Cadherin. Seen this cadherin protein is a cell adhesion molecules. So there is a primary protein called cadherin. Cadherin is then attached to a linker protein. This linker protein is called beta cadherin. This linker protein cadherin is further attached to a cytoskeletal filament, which is actin protein. So this cadherin primary protein is attached to a cadherin linker protein, further attached to a cytoskeletal filament, in this case which is actin. So we've seen previously this molecular hijacking by this microorganism Listeria. This Listeria hijacks this actin polymerization phase of cellular movement, Listeria. This Listeria, how does it does? It contains protein called internalin A. This protein is called internalin A. This internalin goes and attach to cadherin, which is a primary protein seen above in this arrangement. This Listeria microorganism contains internalin A, which goes and attached to the cadherin. This leads to invasion of Listeria. So this invasion of Listeria, you have seen how it does by molecular hijacking, by stopping the actin polymerization. This actin polymerization is stopped by internalin A, which goes and attaches to this cadherin. This is the primary molecule. So we've seen this zona adherence, how they are attached, arrangement, and how they are hijacked by this Listeria organism. We are going to see something in these epithelial cells. There are also called desmosomes. You must be familiar with this word, desmosomes, in the epithelial cells. These desmosomes are present in cells in huge mechanical stress. So the features of these desmosomes are present in the cells in huge mechanical stress. For example, heart cells, skin cells, uterine cervix cells, etc. Uterine cervix cells, skin and heart cells, they present this zona adherens form, special form called desmosomes. So we are going to see their arrangement. First, there are primary protein. Here the primary protein is desmostane. Primary protein are desmostane and desmocolin. These two are primary proteins in the desmosomes, desmostane and desmocolin. These two are attached to the linker protein called desmocollin. desmoplakin. They are attached to the desmoplakin, that is the linker protein, which is finally attached to a cytoskeletal filaments. Example here are the tonofilaments. Tonofilaments are an example of the cytoskeletal filaments, to which this desmoplakin, an integral protein attached to this desmoplakin, this desmostain and desmocolin are attached. This is the arrangement of 3 proteins in the desmosomes. So this autoantibodies against desmostain. So autoantibodies against desmostain leads to a condition called pemphigus. This condition of pemphigus is due to autoantibodies against this desmostain. Desmostain is a primary protein in this arrangement of desmosomes, attached to desmoplakin, further attached to the tonofilaments, that are the cytoskeletal filaments. So this condition of autoantibodies against desmostain is called pemphigus. So we have seen this first type of cell to cell adhesion, zona adherence. Second we are going to see is tight junctions. Tight junctions. So these tight junctions are called zona occludens. Zona occludens. The cell to cell first adhesion are called zona adherence. This tight junction is called zona occludens. The proteins involved here are, in this zona occludens, occludin, very simple, occludin, and claudin protein, occludin protein and claudin protein involved in the tight junctions called zona occludens. These tight junctions are present in 3 important areas. In the first is the blood-brain barrier, in this brain, the blood-brain barrier. Second is the gastrointestinal tract, the GIT. Third important is the kidneys. Tight junctions are present in the blood-brain barrier. We are going to see further the central nervous system discussion, how this blood-brain barrier is formed with the help of tight junction, and also the astrocytes. Astrocytes we have discussed above. So astrocytes and tight junctions help in the formation of blood barrier. We are going to discuss in more detail. They are also present in the GIT and also in the kidneys. So I'm going to see some types of movements here. So this is cell 1 and this is cell 2. First cell, this is the second cell. So here we have 2 types of movements. This first movement across through the cell is called transcellular pathway. This movement is called transcellular pathway. This movement is through one cell, across one cell. And then the second type of movement is through this across 2 cells. The side movement. Side movement is called as paracellular pathway. So across the 2 cells, the side movement is called as paracellular pathway. Across one cell is called as transcellular pathway. We are going to see clinical feature here. This protein called claudin 16 protein. Seen this claudin protein is involved in this tight junction formation of zona occludin and claudin protein. This claudin 16 protein mutation, claudin 16 protein mutation going to affect the transcellular pathways. Transcellular pathways affected by claudin 16 protein mutation, where in the kidneys is affected, leading to this very important condition called familial hypomagnesemia, hypercalciuria, familial hypomagnesemia, hypercalciuria. Magnesium is decreased, calcium is increased. Hypomagnesemia, hypercalciuria, familial hypomagnesemia, hypercalciuria due to claudin 16 protein mutation in the kidneys. As a result, these patients are more prone to renal stones called kidneys are involved in kidney stones are general calcium, majority of the calcium oxalates, nephrocalcinosis. So the kidneys are more susceptible to stones, renal stones, so nephrocalcinosis. This we have to remember. Claudin 16 protein mutation leads to familial hypomagnesic hypercalciuria. So this we have discussed about the tight junction, how this claudin 16 protein mutation leads to nephrocalcinosis. Third one we are going to discuss is the gap junction. First one was zona adherens, second one was the zona occludens. This gap junction doesn't have a very specific name. This gap junction is very much required for rapid communication. So very simple and basic understanding. We know this gap junction is required for rapid communication for transport of various molecules, ions, substances across the cells. So these gap junctions are present in heart. These gap junctions are present in heart. We're going to see in cardiovascular system how they are important. Then in the smooth muscles, where the vascular system and GIT. In the vascular system and GIT smooth muscles, gap junctions are present. In heart, in smooth muscles, third are the neurons. The neurons, these gap junctions are also present. So these gap junctions are found, and to see the protein, these are formed by connexin proteins. Simple and easy to remember. Gap junction, connexin proteins. This tight junction, zona occludin along with claudin. This zona adherens is cadherins, integrins and selectins. So we need to remember one or two out of three, and the others rest will fall in line. Proteins involved in these junctions are important, asked frequently. So this connexin protein is involved in gap junction formation. So we're going to see some of these proteins, how they are affected. First is this CX32 protein. CX32 protein is present in the neurons or the nerve fibers. So when it's affected, these are affected. The nerve fibers or the neurons are affected. It's a type of this disease is called Charcot-Marie-Tooth disease. Charcot-Marie-Tooth disease is due to mutation in the CX32 connexin protein in the neurons or the nerve fibers. It's an X-linked disorder, but this Charcot-Marie-Tooth disease. Next protein we are going to see is CX40. CX40 is present in the heart. Present in the heart is affected. CX40, the heart is affected. CX32 neurons are affected. So in this heart affected, it leads to idiopathic atrial fibrillation. This leads to idiopathic atrial fibrillation, which is due to mutation of CX40 connexin protein. And there is a third protein, CX37. CX37 protein is present in blood vessels, in the small muscles of blood vessels, as you have seen above. This blood vessels, the smooth muscles are affected. Smooth muscles are affected. This leads to a condition arteriosclerosis, a common disorder of the blood vessels. CX37 proteins affected. So CX32 neurons, Charcot-Marie-Tooth disease, CX40 in the heart, idiopathic atrial fibrillation, and CX37 in the smooth muscles of the blood vessels, arteriosclerosis. With this, we have seen all the three types of cell-to-cell junctions. Next, we are going to see is second type of junction, cell basal lamina junctions. There's not much to discuss here in the cell lamina junctions. Remember, there are two types. First one is the hemidesmosomes. The desmosomes were in the cell-to-cell junctions. These hemidesmosomes are in the cell basal lamina junctions. Second one is the focal adhesion. Focal adhesion and hemidesmosomes. These are the examples of cell basal lamina junctions. These hemidesmosomes are attached to intermediate filaments. These hemidesmosomes are attached to the intermediate filaments. This focal adhesion is attached to actin. So this focal adhesion is attached to this actin protein. Hemidesmosomes is attached to intermediate filaments. So disruption of these two junctions leads to cells movement or movement of these cells, leading to an important phenomenon, metastasis. This metastasis is an important clinical phenomenon in the cancer and the carcinomas. The cancer cells travel or they get transported from site of the cancer injury or infection to another, where they could start another dangerous tumor in another part of the body. So disruption of these two junctions, the hemidesmosomes and cell focal adhesion, leads to cell movements, unrequired, non-needed cell movements, leads to metastasis. So we're going to see off topic here. There are marker enzymes for cell parts. It's very important topic for discussion. There are marker enzymes for cell parts for identification. For the identification of the cell parts, there are the marker enzymes. We're going to see some of the cell parts and the microorganisms. First one is the cell membrane. Cell membrane, the marker enzyme is sodium potassium ATPase. Sodium potassium ATPase is present across the cell membrane, so this is a marker enzyme. Another we need to remember is adenylyl cyclase. Adenylyl cyclase and sodium potassium ATPase are marker enzymes of the cell membrane. Next is the endoplasmic reticulum. For endoplasmic reticulum, it is glucose 6 phosphate. So glucose 6 phosphate is a marker enzyme for endoplasmic reticulum. Third is Golgi apparatus. For Golgi apparatus, we have galactosyl transferase. Galactosyl transferase for Golgi apparatus. Fourth is the lysosomes. Lysosomes, the marker enzymes are acid phosphatase and another important cathepsins. Lysosomes, easily remember. We discussed the inside of the lysosomes get acid, so the enzymes are known as acidic hydrolase and acidic proteases. So this acid phosphatase, the acid word we can remember, acid phosphatase along with the cathepsins are marker enzymes for lysosomes. Fifth, we are going to see peroxisomes. Peroxisomes, we have catalase. It's important enzymes we have discussed, along with uric acid oxidase. We have seen this catalase enzyme is important in the scavenging free radicals. So for peroxisome is easy, remember catalase along with the uric acid oxidase. Sixth, we have mitochondria. The mitochondria, the marker enzymes are ATP synthase. Very simple, the ATP synthase enzyme is present in the mitochondria for synthesis of ATP. So ATP synthesis is a marker enzyme for mitochondria. Another one is creatine kinase. Creatine kinase is an important enzyme in the formation of creatine and creatinine. Remember, these two are important enzymes for formation of energy releasing molecules. So this mitochondria, which is the powerhouse of these stores, is marker enzymes are ATP synthase and creatine kinase, the energy releasing molecules. Last one is the nucleus. For nucleus, we have the marker enzyme as RNA polymerase. It's simple to understand. RNA polymerase, DNA RNA present in the nucleus, so RNA polymerase and histone deacetylase. Histone proteins are combined with nucleus to form the nucleosomes, so this is also a very easy. Remember, histone acetylase and RNA polymerase. So it's a very important discussion. I'm going to require this in the pathology subject in the second year. For this, for now we have to remember this marker enzymes, very important. So this concludes today's discussion of cytoskeletal filaments and cellular junctions. Please do revise them, guys. Very important topic. Lots of clinical knowledge is given here, which will be always useful in subsequent years, subsequent subjects, especially in the second year and in general medicine, also in surgery. So make sure to revise them, guys. Thanks for watching. I will catch you in the next one. Good luck.